On my side of the world, it's currently morning. So good morning from a sunny spring day in South Africa. I'm still coming to terms with the fact that we can't all be sitting together in a room today having our usual in-person discussions about our work, but I'm at least grateful that technology has enabled us to come together virtually. I've really tried to make this talk as interesting and as different as possible so that I don't lose your attention to all the home distractions. Don't worry, I know all about those. Before I begin my talk today, I would like to acknowledge the several institutions that played a role in the story I'm about to tell you. All the institutions shown here played some role in my career development, whether through training, contributing financially, or by donating or loaning equipment. Without these institutions, and of course all the people involved, this work would not have been possible.
For those of you that don't know me, I thought I'd introduce myself somewhat personally. My name is Carla Edworthy, and I'm a recent PhD graduate from Rhodes University in South Africa. And I'm currently a postdoctoral research fellow at the South African Institute for Aquatic Biodiversity. I live in this beautiful coastal town on the south coast of South Africa called Port Elizabeth. It's also called the Friendly City and the Windy City, because as you can see, it is almost always windy here. Either way, we enjoy a great lifestyle, which is really laid back, and it's centered around our beautiful oceans, our beaches, and we enjoy all the privileges that a small coastal town has to offer.
Port Elizabeth is situated in a large coastal embayment called Algoa Bay. The bay is known for its incredibly diverse marine life, ranging from marine mammals such as whales and dolphins to seabirds, fishes, corals, invertebrates, plankton, you name it, we have it. The bay is approximately 80 kilometers wide and relatively shallow, but very dynamic in terms of its oceanography, and I'll tell you why. Although this may appear to be a nice and calm little bay, there's actually nothing calm about it. It's a really dynamic coastal area that is influenced by several coastal processes. For example, flowing only 60 kilometers from the mouth, we have the notoriously strong and warm Agulhas Current. This current originates from the tropical east coast of the country, and it carries warm water in a southwesterly direction past the bay. The current is known to retroflect eddies of warm water that enter the bay occasionally, and this depends on the direction of those famous windy city winds that I spoke about earlier. Then we also have several rivers and estuaries, as well as a groundwater seep that provides fresh water inflow into the bay. We also have another interesting feature on the western boundary of the bay, which is a large macroalgal reef. And this reef, interestingly, plays a big role in the pH dynamics of the bay, which I'll speak about a little bit later. Then, of course, being in an urbanized area, we have the influence of industrial processes and also some wastewater treatment plants that eject wastewater into our coastal areas. And being situated on the temperate south coast of the country, and due to the general windiness of this region, the bay also experiences seasonal upwelling, which usually occurs at the boundaries of the bay, and cold upwelled water enters the bay, also depending on the wind direction. And lastly, the more exposed section of the bay, which is called the windward side, because of the predominant southwesterly winds which occur here, there are several currents and mixing processes which influence the oceanography on that side of the bay. So as you can see, this image paints quite an exciting and dynamic picture for the bay's oceanography. Because of how dynamic and, dare I say, exciting Algoa Bay is, just over 10 years ago it was identified as a sentinel site for oceanographic and biological monitoring in South Africa. And the bay has since become one of the best monitored sites in the country. The long-term continuous monitoring program that has been operating there for several years is hosted by the South African Environmental Observation Network. And the reason why this is important will become evident later in my talk. At this point, you might be wondering why I'm telling you all of this. And it's because this particular study site and its existing marine infrastructure provided us the great opportunity to do some awesome research. And yes, I will be getting into my actual ocean acidification research now, for those of you that have been wondering when the subject is going to come up. I'm sure many of you are familiar with this publication by some of our favorite GOA-ON colleagues. This paper highlighted where the bulk of ocean acidification research originates from in the world, and also where it is lacking. As you can see, there is a general lack of published ocean acidification research from Africa. This picture would have changed quite a bit by now, thanks to the efforts of GOA-ON and its several partners over the last few years to build capacity for ocean acidification research. However, we in Africa still have some catching up to do in terms of improving our understanding of ocean acidification in our local waters. As many of you will know, there has been a recent drive in our scientific community to understand local ocean acidification conditions and impacts, and this is in order to target our response and promote adaptation to future changes. And that is exactly what the overarching goal was of my study. So with all of this information in hand about how interesting and important my study site was, the current monitoring efforts that were already taking place there, and the lack of information on ocean acidification, specifically coastal acidification in Africa, I knew I wanted to start monitoring the local pH conditions and to contribute this information to the ongoing long-term monitoring already taking place. Despite having this wonderful infrastructure in place, with access to boats, CTDs, data, we were not at all equipped to measure pH and carbonate chemistry reliably. In fact, the CTD's pH sensor had been giving strange readings for some time, and everyone really wanted to have a better understanding of what was going on. Fortunately, a few years prior to this, I by chance applied to attend an introductory ocean acidification course in Mauritius, hosted by the Ocean Foundation and GOA-ON and their institutional partners. This course was really where I started networking with the ocean acidification community, and after applying, I was lucky enough to be awarded the GOA-ON OA in a Box kit, which was exactly what we needed to kick off our ocean acidification campaign here in South Africa. The GOA-ON kit included everything that was needed to implement a simple monitoring strategy and to measure pH and total alkalinity from water samples back in the laboratory, and also everything that was needed to conduct biological experiments with pH control. The kit also included an ISAMI pH sensor for continuous pH monitoring, and this proved to be really useful for capturing local variability, and I'll show you some of this data later in the presentation. My research goals were not only to monitor pH, but also its impact on a socio-economically important marine fish species. So my starting point was to come up with a relevant research question, because as we all know, all good science starts with a good question. So I set out to identify what it was that I wanted to know, why I wanted to know it, and how I could collect information to answer these questions. The major research question was to identify how pH varies locally and how this influences a fish species who occurs in the most variable coastal habitats. My next step after formulating my question was to evaluate what resources were available to me so I could find the best way to collect the data that I needed. And this just happened to become a valuable lesson on where and how to start when developing a new program like this. So I set out to put Algoa Bay on the map for coastal pH monitoring by sampling a series of 8 offshore and 5 inshore stations in the bay on a monthly basis over approximately 20 months. This coincided with the long-term monitoring program's monthly trips, which gave me access to boats, CTD data, vehicles, and of course manpower. At each site, I collected a water sample following the standard sample collection, standard operating procedures for pH and total alkalinity measurements back in the lab. In addition to our monthly sampling, we also deployed the ISAMI pH sensor at the macroalgal reef site in the bay. It was deployed in a shallow gully, secured in a steel cage, and a danger sign on it to deter any sticky fingers. The sensor measured pH continuously every 30 minutes over 5 consecutive days. We selected this particular site for deployment, mainly because it was shallow and calm, so that we'd avoid the sensor being lost in rough seas, but also because it's a known nursery area for young fishes. and we knew that our study species occurred here in abundance. This area is also security protected, so it was somewhat protected from theft, which was my biggest fear for the RSAMI pH sensor.
This site right here was the most interesting in terms of pH variability during my study, although it looks rather unsuspecting, doesn't it? Not only did my study species occur here in high abundance, but this site also showed some really interesting pH dynamics, and I'll be discussing those results in the next slide. This was also the site where we deployed the RSAMI pH sensor for a period of 5 days to look at diurnal variability in pH.
Although the monthly monitoring revealed some fairly interesting spatial and temporal patterns in pH, I've chosen to focus on the data produced by the continuous monitoring by the RSAMI pH sensor in this talk. And that's because it revealed the highest variability likely to be experienced by our study species. The RSAMI pH sensor revealed a really clear cyclical change in pH over 24 hours, as you can see in the graph on the left. pH rose to a high during the day, peaking around midday and dropping significantly at nighttime. And interestingly, the peak pH during the day reached levels as high as 8.4. This data was indicative of potential biologically mediated pH, and this would make sense considering the large macroalgal reef at the site. And this is definitely something that we intend on exploring further in future research. And this would be interesting to pursue, considering that this area seems to serve as a nursery for young fishes, and due to the high pH levels during the day, it may also serve as a potential refuge to ocean acidification for the organisms living there. The ultimate conclusion from this part of the study was that using continuous pH monitoring was better at capturing local variability, which has given us some insight on where to focus our efforts for future research. Like I mentioned earlier, I'm also presenting the results from a biological component to my research. Enter the blacktail, a common coastal fisheries species in South Africa that is named after the black spot on its tail. As you can see, even the tiny post-flexion stage larvae, shown in the picture in the center of the screen, the black spot is easily visible, making the species really nice and easy to identify in the field. This species is abundant in our coastal waters and has a really flexible life history strategy. It occurs as larvae and juveniles in almost all coastal habitats, including rock pools, surf zones, estuaries, and it's most frequently caught in our subsistence and recreational fisheries. This was a perfect species to study for several reasons, the most important being that it was accessible for the required numbers for our experiments, and it's easy to keep in the laboratory. Its broad distribution and ability to live in harsh environments, as well as its popularity in fisheries, made it a perfect species to assess. We conducted physiological and behavioral experiments on early-stage blacktail, and the results showed that this species could tolerate pH levels as low as 7.2, with no physiological changes evident and only subtle changes in activity levels in response to the low pH treatments. This was somewhat expected, considering that the species is known to be exposed to high pH variability based on its occurrence in those dynamic coastal habitats that I mentioned in the last slide. However, this scenario is not true for another coastal species, because a previous study on a coastal and estuarine species called the dusky cob revealed that this species had high sensitivity to pH levels of only 7.78, and this is despite its really similar coastal distribution to the blacktail. And this really highlighted the importance of identifying species-specific responses to low pH for important fishery species. In a way, my study served as a trial and error attempt to initiate a monitoring program for ocean acidification in a region where such data has not yet been collected. And doing this, we were faced with several constraints and limitations, but also some opportunities. Identifying these were integral to the success of this research. My mentor and supervisor, who many of you probably know, Professor Sam DuPont, taught me one valuable lesson which I'll carry with me forever, and that was to always do the best you can with what you have. By identifying what we have, which in my case was an existing program, access to infrastructure, support and easy access to an interesting study species, we could use this information as a starting point for our research. And of course, I realize that not everyone is privileged to have this many opportunities, but even just an interesting question or a potential contact you make here at OA Week can become an opportunity to start something new. And of course, we also had several constraints, which may have hindered our progress somewhat, but ultimately you make it work to the best of your ability. After all, there is no such thing as science without limitations. I would also like to add my own quote at this point, which is, the best way to start something new is simply to start. To summarize, one of the outcomes of this research was the development of these simple guidelines to developing a successful and simple monitoring program that assesses species responses in the context of local conditions. The first step being to formulate a strong and relevant research question, then to identify a suitable study species that has some socio-economic relevance and is also accessible in the required numbers for experiments, identifying a suitable study site and a method for monitoring that will capture the variability that your study species is naturally exposed to, and identifying suitable experimental treatment levels to test the hypothesis that the study species is adapted to manage the pH levels within the range it's naturally exposed to. We are not planning on stopping here with our research on ocean acidification. Now that my ocean acidification monitoring has had some success and gained momentum, this has sparked the interest of other organizations and researchers who are investing in future research on the subject. For example, the ongoing long-term monitoring program that I keep speaking about has added pH and total alkalinity measurements as permanent recordings in future, and they are invested in training other staff and students to measure pH and analyze these data. We have also invested in 8 ISAMI pH and PCO2 sensors to add to our offshore moorings throughout the country in order to better understand the conditions in our coastal regions. As mentioned earlier, we are also planning to investigate how biologically mediated conditions work in vegetated nursery habitats, including the macroalgal reef, as well as seagrass habitats. This just shows that a little effort and a little passion goes a long way in attracting attention, funding, and interest for topics like ocean acidification, and the only way to get there is to start somewhere. I also just wanted to mention another aspect of my research, which has been quite successful at getting information about ocean acidification out into the public. I've always had a vested interest in doing research that is meaningful to people and finding creative ways to share our ideas in science with society. Firstly, the monitoring data from the study has been submitted to the Sustainable Development Goals Target 14.3 data portal, and we've used several science communication platforms to share our work, such as social media, popular articles, and national and international science events. Our Ocean Acidification Day event on World Oceans Day in 2017 was covered extensively by national media and radio, and this was an exciting way for us to get our message out there about ocean acidification and its impacts.
That concludes my story about my ocean acidification research in South Africa, and I would like to thank you all again personally for listening today. I hope that my talk gave some of you who haven't done any ocean acidification research in your region or country some ideas to start. Or for those of you who have been researching ocean acidification, I hope this gives you some new perspectives